be a little bit easier because you can actually watch it from your phone if you have the YouTube app. So, all right, so I've got it recording now. So we're going to look at chapter 19. So let me share my notes. And we will get going. Okay, so we're going to be looking at the reproductive system this week. And as you learned from the video and listened to Hank Green talk about the reproductive system, you know, all of our other systems have been keeping us alive. This one is a little bit different because it's not keeping us personally alive, but being able to keep the species alive. So being able to allow for the reproduction so that we can produce new humans, so we can keep our species alive. So we're going to look first at kind of some basic structures. So just some terminology as we get into the reproductive system. And I'm starting on page 640 in your book if you want to follow along in the book. The gonads, now the gonads are the actual reproductive structures where the reproductive cells are produced. So for the males, that's the testes, and for the females, that's the ovaries. So that the gonads are the places where we actually get the sex cells, the sperm and the eggs. And then we have ducts, and those are going to vary. The male has a much more extensive duct system than the females. We've got some accessory glands, the majority of those in the male. And then we have the external genitalia, okay? And so those are, of course, going to be in the perineal region. So we're talking about the penis for males and the labia majora and labia minora for females. So the reproductive cells, or the gametes, are produced in the gonads, and the sperm in males, and then the oocyte in the females. I know that looks like it should be pronounced oocyte, but it is oocyte, and that's the technical terminology for the egg in females. And with your vocabulary, you know that the root OON refers to an egg, and so oocyte means egg cell. Okay. So the oocyte is actually an immature gamete, and it can't continue dividing until fertilization occurs, and we'll get to that a little bit later on. All right, so now we're going to start with the male. So we're going to be on 641, and we're going to look at the different structures of the male reproductive system and kind of what they do. So first of all, the male reproductive system, one of the major differences between the male and female is that part of the male reproductive system is enclosed in the scrotum outside of the body. Okay, so the scrotum is where the testes are found, hangs outside of the pelvic cavity, so it's external to the pelvic cavity. It's not going to be inside the pelvic cavity like the female organs. So the testes, or the gonads, are also referred to as the primary sex organs, are found in the scrotum, and both testes would be found there, each one in a scrotal cavity. Okay. The testes are about 2 inches long, about a little over an inch wide, and an inch thick. They weigh about half an ounce a piece or so. They're roughly the size of a flattened egg. Okay. Within the wall of the scrotal cavity, there is a serous membrane, and remember the purpose of the serous membrane is to reduce friction. So there is a serous membrane between the cavity wall and the testes, so that there's little friction that's created there. The scrotum consists of a very thin layer of skin with some smooth muscles. And there's two different muscles, one being a smooth muscle, and it's a whole layer, and that's called the dartos muscle. And the dartos muscle is what causes the wrinkling of the scrotal surface. But there's also another layer of muscle called the cremaster muscle, which contracts to actually pull the testes closer to the body to make sure that we have the proper temperature for sperm development. And the reason why the testes are outside of the body, as you learned in your video, is that body temperature is actually too warm for sperm production. Temperature that's about 2 degrees Fahrenheit cooler than normal body temperature. So by having the testes located outside of the body cavity, then we can have that cooler temperature. If air temperature or body temperature rises, then the cremaster muscle can relax and the testes will move away from the body. Cooling of the scrotum would cause contraction of the cremaster muscle, which would pull the testes in closer to the body. So the testes, because they are outside the body and they don't have the normal protection that our internal organs have a very thick, fibrous capsule that surrounds them called the tunica albuginea. And the tunica albuginea folds so that it can divide the testes into different lobules. So the tunica albuginea, this really thick fibrous tissue, divides the testes into about 250 lobules. So within each testis, we're going to find lobules, and within each one of those lobules, we're going to find the seminiferous tubules. And these seminiferous tubules are tightly coiled within each of these lobules, and altogether, the male has about a mile of these seminiferous tubules. If we were to take them out and stretch them out, it would equal about a mile of tubules. Okay, and it is in these seminiferous tubules that we have sperm production. Okay. Now, in between the tubules, we have the interstitial cells. And these interstitial cells actually produce testosterone. So the cells within the tubules produce the sperm, and the cells in between the tubules, the interstitial cells, produce testosterone. And the sperm, once they are produced in these seminiferous tubules, then leave the tubules through the rete testis and the efferent ductules, and then get into the epididymis, which really is the first part of the male reproductive tract, really the first part of those ducts that are going to lead ultimately to the urethra, so that the sperm can be deposited outside of the body. So here you can see a horizontal section through the scrotum and the testis. So now you can see the skin. Again, it's a very thin layer of skin there. You see your dartos muscle and the cremaster muscle, and then the cavity, and then the tunica albuginea is what actually surrounds it with those lobules. Now in this, again, it's only showing 5 or 6 lobules there, but keep in mind that there would be about 250 of these little lobules within the testis. And these little coils that you see here in purple, those are your seminiferous tubules. And this little network here that kind of looks like a capillary bed is actually the rete testis. And so and what it's not showing here is actually all of these would be connected to the rete testis. And then they're going to leave through the efferent ductules and get into the epididymis here, which is kind of a comma-shaped structure that we see there. So your slide, and you'll look at a more detailed picture of this for your lab, but your slide of the seminiferous tubule will look something like this. Each one of these coiled purple structures here is a section through a seminiferous tubule. And so some of the views that you're going to get will be a perfect cross section, like this one that they've got kind of pointed out here right in the middle. These that look similar to this is more of a cross section. And then you've got these that are elongated, these two kind of over to the side here. And this would be more of a longitudinal section. So you're going to get all different types of sections through the seminiferous tubules, depending on the cut that was made. What you'll see also are your interstitial cells. So all of these little cells hanging out in these white spaces between the seminiferous tubules are going to be the interstitial cells. And if we take a closer look, if we look over here at this figure C here, what we're going to see are the different types of cells that are found within the seminiferous tubules. Okay. And then we'll see some of the spermatids and then actually some of the spermatozoa. And you'll see those when you look closely at these slides at a much higher magnification. You see this is just times 75. But if we look at a higher magnification here, you'll see some of these spermatids and some of the spermatozoa. You'll also see what we call these nurse cells or the sustentacular cells. The sustentacular cells are there to help provide nourishment for these developing sperm cells. So the process of sperm development is called spermatogenesis. Okay, and spermatogenesis takes place in these seminiferous tubules, and it is a series of cell divisions that take place with mitosis and meiosis. And what happens is each successive division produces these daughter cells that go toward the lumen. Okay, let me go back. Oops. All right. So remember, the opening is called a lumen. Okay, and it doesn't really matter what we're talking about, any hollow structure, the lumen. So we're going to have division that takes place, and each successive division is going to push the daughter cells toward the lumen. So eventually we end up with the spermatids and the spermatozoa in the lumen. Okay. All right, so the first thing that we're going to get is mitosis. And remember that mitosis is the type of cell division that we have covered before. Okay, we talked about mitosis. I know it's been a long time, but we talked about that all the way back in chapter 3. Okay, so if you go back to chapter 3, if you flip back in your book to chapter 3, pages 84 and 85 show us the stages of mitosis. And remember, we looked at these, we looked at those dividing cells, and you had to find the cells that were in each stage and them throughout. So that's mitosis, where the daughter cells contain the same number of chromosomes as the parent cell. So let me get back here. All right. So mitosis is what I'm going to refer to here as normal cell division. Okay, where we get the parent cell divides into two genetically identical daughter cells. Okay, so mitosis is when our parent cell divides into two genetically identical daughter cells. Okay, now these genetically identical daughter cells, if we look at the genetic material, okay, they contain 23 pairs or 46 chromosomes. And that is the number that we have. We have 22, what we refer to as autosomes, okay, and or 23 pairs of autosomes, and one pair of sex chromosomes, which determine whether we are biologically male or biologically female. Okay, so we have 22 pairs of autosomes and one pair of sex chromosomes. These types of cells are referred to as diploid or diploid cells because they have two copies of each chromosome. Okay, they have two copies of each chromosome. So that is our first step in spermatogenesis. Okay. So spermatogenesis step one is mitosis. Okay. And what happens is we have these stem cells called spermatogonia. Singular would be spermatogonium. Okay. So we have these stem cells called spermatogonia located in the outermost layer of the seminiferous tubule. And they just undergo meiosis throughout their entire life. Okay. So throughout their entire life, they undergo mitosis. Okay. One of these daughter cells, we're going to call them daughter cells, remains in place. It remains there in place as a spermatogonia. Okay. And the other daughter cell moves closer to the lumen and becomes what we call the primary spermatocyte. Okay, so these spermatogonia undergo mitosis throughout their entire life, and since they're producing these genetically identical daughter cells, one of those daughter cells is going to remain in place, remain a spermatogonium, so it can continue undergoing mitosis. The other moves a little bit closer to the lumen, moves kind of to the next layer of cells, and forms this primary spermatocyte. Okay, and then we have another round of mitosis. Okay, and they differentiate then into the secondary spermatocytes. Okay, so the primary, oops, oh my goodness, okay, I cannot type tonight. Primary spermatocytes undergo mitosis again to become secondary spermatocytes. Okay. Oh, okay. Your book doesn't really talk about this in these. Hold on. Let me erase that. Don't write that. If you've written that, erase it. Sorry. The primary spermatocytes undergo meiosis. Sorry. To become secondary spermatocytes. Okay. So this is step 2. Now, meiosis is different. In mitosis, we're producing genetically identical daughter cells. In meiosis, or sometimes this is referred to as meiosis, we are producing 4 genetically different daughter cells. Okay, so meiosis actually has 2 different stages. Okay, we have meiosis 1. Okay, so that's meiosis 1. And in meiosis 1, in the first cycle, we produce 2 cells. So we still only get 2 cells after the first step. And these are called secondary spermatocytes. Okay, these are called secondary spermatocytes. So after the first round of meiosis, the first cycle of meiosis, we get 2 cells called secondary spermatocytes. Basically what happens is we get some exchange of genetic information that can occur that we don't see with mitosis. The secondary spermatocytes contain 23 chromosomes. They are no longer in pairs. Okay, so essentially what happens is that each one of these cells now has only half of the genetic material. Okay, so these cells are haploid. Okay, because they contain essentially half or only one set so they contain half or only one set of the genetic material. Okay, and then these 2 cells, these secondary spermatocytes, then undergo the second part of meiosis. So they undergo meiosis 2, okay, the next cycle, to produce 4 cells that are genetically different and all haploid. Okay, so they all only contain half of the genetic material, just one set, because they're going to fertilize the egg. The egg contains one set or half of the genetic material. So when we put those 2 together, we have a whole set of genetic material again. Okay, so the secondary spermatocytes undergo meiosis 2 to produce the 4 cells that are genetically different and all haploid, and these are called spermatids. Okay, so these are called spermatids. Okay, and then kind of the last step. So this would be step 3, essentially. And then the last step is called spermiogenesis. So a little bit different, spermiogenesis. And this is just maturation of the sperm. Okay, so at this point in step 3, they are not mature. So step 4 is actually when the sperm mature. That's the spermiogenesis part. Okay, so if we look in the book on page 644, okay, so this is figure 19.3 on 644. So here is our spermatogonium at the very top. Okay, it undergoes mitosis, and one of those stays there. The other becomes the primary spermatocyte. Okay, that's diploid. Remember, it's going to have 2 copies. All right. Then it undergoes meiosis 1 to form the secondary spermatocytes that are now haploid. Okay, and we get the spermatids haploid, and then that then mature through the process of spermiogenesis to become spermatozoa. So basically, what this means is that males, once they hit puberty and once this process begins, they can produce sperm forever. Okay, as long as they have these spermatogonium cells, then they can continue to produce sperm. That's why, 85-year-old men can father children, but 85-year-old women cannot have children because women go through menopause and we stop producing eggs. But men never stop producing sperm. Now, the quality and the quantity of the sperm may decline, but as long as they have spermatogonium cells, they can continue to produce. Okay. So when we look at a spermatozoan, and of course we call these sperm for short, but their technical term is spermatozoan. So when we look at a spermatozoan, we have 3 distinct portions: the head, the midpiece, and the tail. Okay, the head, the midpiece, and the tail. So in the head region, we find the nucleus, okay, and then we find this tip called the acrosome, and the acrosome is going to have enzymes that are going to allow it to dissolve the outer structure of the oocyte and be able to get into it to cause fertilization. Okay, in the midpiece, okay, in this midpiece region here, we have lots and lots of mitochondria. Okay, so really, in terms of organelles, the sperm cells are a lot different than normal cells. We basically find a nucleus, some centrioles, and then a ton of mitochondria. And the mitochondria provide the energy to fuel the tail. Okay, and the tail is responsible, for being able to propel the sperm up through the female reproductive tract. Okay, so the mitochondria are going to allow the sperm to be able to move. Kaylee, is your screen still blank? I'm sharing my PowerPoint. Okay, good. So these mitochondria provide the ATP for the flagellum, and they're going to be able, the cells are going to be able to get some nutrients from semen. So it's going to provide the necessary sugar for these mitochondria to be able to produce ATP. So if we look at the structure of the male reproductive tract, this image is back on page 641. So this is our sagittal section. of the male reproductive system. So we can see the testes in the scrotum outside of the pelvic cavity. All right. You see the epididymis. I don't know why the lines aren't showing up real well, but the epididymis is this kind of comma-shaped structure that sits behind or posterior to the testis. Then we've got this long duct that goes up from the epididymis up through the scrotum back into the pelvic cavity and kind of coils around behind the bladder. That is the ductus deferens or the vas deferens. Okay. And then we've got some glands. We've got the seminal gland that sits posterior to the bladder. We have the prostate gland that sits kind of inferior to the bladder. And then we've got these tiny, tiny little glands inferior to the prostate gland called the bulbourethral glands. Okay. And all of these glands are going to make different secretions that combine with the sperm to produce semen, and then the duct becomes the urethra, and that's what finally is going to be, it's going to allow the sperm to leave the body. All right, so now kind of looking at this tube structure here, or the duct structures, as we go through the male reproductive tract. Okay, so first we've got the epididymis, and the biggest thing here about the epididymis is that this is where the sperm go to mature. They're going to take a long time to go through the epididymis, and they are going to mature here. It takes about 2 weeks, and so they leave the tubules really as the sperm cells that aren't mature. They can't do anything at that point. But in their journey through the epididymis, this is when they mature. So they detach from the nurse cells. They sit in the lumen of the seminiferous tubule, and they have the characteristics of the mature sperm, but they're still functionally immature. They can't coordinate locomotion, and they're not capable of fertilization at this point. So when we say that they're immature, what that means is that they can't control the flagellum, and they don't have the necessary components to be able to fertilize the oocyte. So they get into the epididymis. It's almost 23 feet long. It's very twisted and coiled, so it takes up very little space within the scrotum. The epididymis also is going to kind of clean up. It recycles the damaged spermatozoa, adjusts the fluid that's produced, and it takes, again, about 2 weeks. So when the sperm leave the epididymis, they are mature, but they are still immobile, and that's going to happen a little bit later, where they actually gain mobility. They gain the use of the flagellum. So once they leave the epididymis, they go into the ductus deferens or the vas deferens, and there is what we refer to as the spermatic cord, which is what allows the vas deferens and some blood vessels to get back up into the pelvic cavity. Passes right beside the urinary bladder and kind of curves around behind or posterior to the bladder. And we have the seminal gland ducts merging with the vas deferens at what we call the ejaculatory duct, which, like I mentioned, goes into the prostate gland and empties into the urethra. So if we look at the structure here, again, this is the posterior view of the bladder. So here's the ureter. Okay, we talked about that in our last unit. So here's the ductus deferens, one from each side. They're going to come around beside the bladder and then wrap around behind. And we've got these 2 seminal glands, again, one on each. And the part where the vas deferens and the seminal gland merge together becomes the ejaculatory duct, and that's what goes into the urethra at the prostate. Now, the way that this looks here, looks like the prostate's actually 2 glands, but we only have the males only have 1 prostate gland, and it encircles the entire urethra, right at the base of the bladder. And so this little portion of the urethra we refer to sometimes as the prostatic urethra because it goes through the prostate gland. And then right after the urethra leaves the prostate gland, we have these tiny little glands on either side called the bulbourethral glands. So another huge difference between the male and female reproductive tract is that the males, sorry, my kids are talking and it's making me lose track. Sorry. Oh. Okay. It's probably cutting in and out a little bit. My internet's not the best, and my kids are talking and they're distracting me. Hang on just a second. I'm back. So the other big difference, or one of the other big differences between the male and female is that the urinary and the reproductive systems both share the urethra, and that's not the case for females. So for males, the urethra carries both urine and semen. Okay. So these accessory glands that we've talked about, the seminal vesicles and the prostate gland and the bulbourethral glands, they produce the fluid component of semen. So semen is composed of sperm and all of these fluids that are going to be produced by these glands. And these secretions are going to do a lot of different things for the sperm. So the seminal glands contribute 60% of all the fluid that is in semen. They're secretory glands. They produce the fructose that is used by the mitochondria. So the mitochondria that are in the midpiece of the sperm do not use glucose. You know, in all of our talk that we've done about the mitochondria, and you know, they're the powerhouse of the cell because they convert glucose to energy. But for the sperm cells, they use fructose instead of glucose, which is another simple sugar, very similar in structure to the glucose. It is more easily metabolized by the spermatozoa. The seminal vesicles also produce prostaglandins. Prostaglandins are kind of similar to hormones. Sometimes they're referred to as tissue hormones. But these particular prostaglandins stimulate smooth muscle contractions along both the male and female reproductive tracts. So for the male, these prostaglandins assist in projecting the sperm through the urethra and out of the male penis. And then for females, they stimulate the smooth muscle in the vagina and the uterus to have contractions to help propel the sperm up toward the fallopian tubes to meet the oocyte. They contain fibrinogen, and fibrinogen helps to essentially clot the semen or cause the semen to become thickened when it's in the vagina. And again, this just gives the sperm more of a chance to be able to travel through the uterus to reach an egg. The other part of this is that it is slightly alkaline. The female vagina tends to be acidic, and under normal circumstances, the sperm really wouldn't be able to survive in that acidic environment. So the seminal glands produces some alkaline substances that help to neutralize and provide a buffer between the sperm and the acidic secretions of the vagina. So now the, these mature sperm undergo what is called capacitation. And capacitation is basically them gaining the ability to swim. So basically at this point now, the tails are going to be motile. They're going to be able to move the tails, and the sperm become motile at this point. The prostate gland forms pretty much the rest of the secretion. The bulbourethral glands, because they're so tiny, they contribute just a tiny, tiny amount of fluid in the semen. But the prostate gland produces this prostatic fluid with a protein called seminal plasmin, and it contains antibiotic properties that appear to help prevent UTIs in males. We know that males experience urinary tract infections at a much lower frequency than females. Part of that's due to the structure of the urethra. The male urethra is much longer than the female urethra. Also because the male urethra is not close to the anus as it is for females, which a lot of UTIs are caused by E. coli, which can be present in feces. So there are some anatomical differences that account for that, but they believe that this particular structure helps to prevent urinary tract infections in males. And there are some other components in there, not really sure exactly what they do. And then the bulbourethral, or the Cowper's glands, they secrete that final amount. Again, it's usually between 1 and 10%. It's a tiny amount that they contribute, but it's mostly mucus. And again, it is alkaline or basic that helps to neutralize the acidic environment of the vagina and helps to lubricate the glands or the tip of the penis. So, semen is comprised of the spermatozoa, the seminal fluid, and these different enzymes. Okay. Each ejaculate, or each time the male ejaculates, the volume of semen is about 2 to 5 milliliters. And we're going to have between 20 and 100 million sperm per milliliter. Okay. And the fewer the sperm, having a lower sperm count could lead to, infertility, or at least problems with fertility. Okay. If there's not that many sperm. The seminal fluid is all of the liquid portions, the glandular secretions, the bulbourethral glands, a very tiny amount of that, the seminal glands producing the majority of that. And then these enzymes, a protease that helps dissolve mucus in the vagina, the seminal plasmin, again, that the antibiotic enzyme that may actually kill bacteria in the vagina as well. And then the enzyme that causes the semen to clot so that it remains in the vagina so the sperm can move through the female reproductive tract. And then an enzyme that actually liquefies it after a time. Okay, so that is the components of semen. So it's not just sperm, it is all of these other substances. So if we take a look at the external genitalia, when we see the male external genitalia, we have the scrotum, which contains the testes, but also the penis. The penis has a dual purpose of being a part of the urinary tract, essentially because of the urethra, but also being able to introduce semen into the vagina during sexual intercourse. So if we look at this image here, this is page 648, we'll see the body or the shaft of the penis. The root of the penis is inside the pelvic cavity. It's the fixed portion that is connected to the body wall. The shaft is where we find the erectile tissue, and then we have the glans, which is the expanded distal portion that contains the what we refer to as the external urethral orifice, which is the opening there. Okay. So within the penis, we have different columns of erectile tissue. We have two columns of corpora cavernosa and one column of the corpus spongiosum. The erectile tissue consists of a lot of vascular channels, so allowing blood flow. There's also some elastic connective tissue and smooth muscle. And during erection, the parasympathetic neurons are going to release nitrous oxide, which is a vasodilator. So that allows those blood vessels to dilate, which allows blood flow to come into the penis, which allows those erectile tissues to become erect and the penis to become hard. Now, when we look at the penis in cross section, and you'll look at an actual slide when you do this lab. Oops. See, I'm not even touching it. What's going on? Okay. So there is, we'll see the corpus spongiosum, just one column of that. It's a smaller column, and it surrounds the urethra. And then the larger columns of erectile tissue are referred to as the corpora cavernosa, and there are two of those surrounding the deep artery. And then these are going to be filled with capillary beds that are going to be able to fill with blood and allow the penis to become erect. So what they're showing you here in this frontal section, you'll notice the vas deferens coming up through here, but also associated with the vas deferens in that spermatic cord would be some blood vessels. Okay, so there would be some blood vessels and some nerves associated with that as well. So if we look at hormones, which we have already talked briefly about these hormones already, but if we look at the hormones of the male reproductive tract or male reproductive system, and there's going to be some overlap between male reproductive hormones and female reproductive hormones. GnRH, FSH, and LH are going to be the same between males and females. Both males and females secrete GnRH, FSH, and LH. Okay, their functions, or the functions for FSH and LH, are different for males and females, but they are still going to be secreted. So GnRH, let me go back here to my whiteboard. So GnRH we talked about already as a releasing hormone, but this is gonadotropin releasing hormone. Remember, our hypothalamus secreted those releasing and inhibiting hormones. So the hypothalamus secretes this GnRH, this gonadotropin releasing hormone. And then this is going to be carried to the pituitary. And so FSH, follicle stimulating hormone, named for actually what it does in the female, but follicle stimulating hormone, and LH, luteinizing hormone, are secreted by the pituitary, anterior pituitary. I wonder why you can't ever see it, Valerie. Can you see it when you watch the videos? No. Is anybody else having trouble seeing it on the whiteboard? Huh. It just lags. And I'm sure that's just connection. Well, I'll tell you what. I'll copy it in the chat. I'll copy what I'm writing on the whiteboard into the chat. That may help. All right. So that may help. So GnRH was the first one. That's the gonadotropin releasing hormone secreted by the hypothalamus. And then that triggers the anterior pituitary to release the follicle stimulating hormone and the luteinizing hormone. Okay. Now, this does not happen until puberty. Okay. So this does not happen until puberty. Okay. Tell you what, I'll just use the chat box to type. That may be the easiest thing to do. All right. So I'll just type in the chat box and leave it there. All right. So this happens in puberty. Okay. So the FSH, the follicle stimulating hormone, targets those nurse cells. And just as a refresher, the nurse cells, also called sustentacular cells, are found in the seminiferous tubules. Okay. And so that triggers the process of spermatogenesis that we looked at before. So that's when our mitosis and meiosis 1 and meiosis 2 are going to start taking place. Okay, that's due to the FSH. The LH acts on those interstitial cells. Remember, the interstitial cells are those in between the seminiferous tubules. Okay, and they start producing testosterone. And when the interstitial cells start producing testosterone, this is when we start to get sexual maturity. So this is when the male starts to develop some of those secondary sex characteristics. Okay. Secondary sex characteristics for males are going to be the development of, chest hair, axillary hair, facial hair. It's going to act on the larynx to trigger the deepening of the voice. It's also going to usually trigger a growth spurt. So the male is going to get taller, get larger, and start to stimulate more muscle development throughout the body. And at this point, the male sexual maturity is initiated. At this point, once the male hits puberty and start this sperm production, then they can start to experience erections and ejaculations. Okay. The testosterone also helps to maintain all of those different glands. So the testosterone helps to maintain the functionality of the prostate gland and the seminal vesicles. It also maintains the libido or the sex drive for the males. And all of these, then as we're starting to get all of this production, the testosterone can feed back to the hypothalamus. And this is shown on page 649. So what happens is that high levels of testosterone feedback to the hypothalamus. Okay. And when that happens, we get the inhibition of the GnRH and therefore FSH, and LH. So the high levels of testosterone inhibit the release of the GnRH, causing a reduction in LH, potentially a reduction in FSH. And the faster the rate of sperm production, the more inhibin is secreted. So we also have something called inhibin. And it is secreted in response to sperm production. So high rates of sperm production lead to the production of inhibin, which also feeds back to the hypothalamus. Okay. So that also feeds back to the hypothalamus and tells it to slow down the release of GnRH, FSH, and therefore LH. All right. So that is the end of the male reproductive system. So what questions do you have about that? Anything? No questions? Okay. Normally at this point, if we were in class, we would go on to the lab on the male reproductive system, which I do have ready for you. So if you wanted, I mean, you've got a class time to work on this, but if you wanted to work on your Chapter 19 lab, I do have it ready for you to do. So if you wanted to work on it. Matter of fact, let me show it to you right quick. Let's see. Hope this works. Yeah. All right, so this is going to be similar to how Lab 18 was set up. I've got some histology videos in there for you. The first three, the testes, the penis, and the sperm, of course, correspond to what we talked about today with the male reproductive tract. And then you've got those histology links, just like before. The first two are pretty much the same as they were last time, where you can enlarge and you can see the different things. The sperm smear is a little bit different. It's not interactive. You'll just draw it as you see it. But the testes and the penis histology are both like they were before. So all of that's up there for you. So if you want to work on that, you could. But otherwise, I'm done with the lecture for tonight. So do y'all have any questions? Nothing? Okay. Well, I will see you back here on Thursday for our lecture on the female reproductive tract. It's a little bit more involved. It probably will take us a little bit longer than with the male. And then I'll try to get the link out to you sooner than 5 minutes before class. And then I'll make those changes in the schedule. I'll prepare my lecture for Chapter 20 to not take quite as long so that we can squeeze in that review on the 21st. So if y'all don't have anything, y'all have a good night, and I will see y'all back here on Thursday. If you think of any questions, just shoot me a text or send me an email, okay? Y'all have a good week. Bye.